We see imaging coming up at several different beamlines. We do see medical experiments at 4max and downmax and Softimax and nanomax. So there is a lot of stuff going on. The portion of scientists coming to Max 4 is really growing, and there is really a potential for further growth. We have come from an area where we really looked at pictures, static pictures. Now with the new technology, we can actually go towards movies.
Maybe I should take the opportunity to say that this is a fantastic place to work in. The beam has a fantastic quality. It's very stable and it has a small focus. We are working on the properties of the pig heart, mainly because it is very close to the human heart, and it has also been potentially a source for heart as a transplantation donor. The focus of our study here at Max 4 was to study the structure of cardiac muscle in a very special situation called the stone heart. When the heart is exposed to severe lack of oxygen, then you can get a very severe contracture that the heart gets into some kind of spasm. We found that there were no evidence for shrinkage at the contractile filament level, and we could measure that by using two very strong scattering reflections.
Universal blood is a type of blood which have been stripped of some specific markers that are recognized by antibodies in recipient blood plasma. We have discovered a set of enzymes that are grouped in two enzyme blends. One of them was able to convert the group B canonical antigen, but additionally an extended structure which has been recently discovered by the group of Martin L. Olson. Similarly, for the A blood group, we have been able to discover an enzyme cocktail of 3 enzymes that were able to handle the canonical A antigen and 3 of the extended structures. These enzymes together enabled us for the very first time to identify the extended structures as important antigens that define the phenotype of the A and the B blood groups. And what this meant from the universal blood type of perspective is that we have now an entry to truly address generating enzymatic universal blood. So these fantastic crystal structures that we have obtained were generated at Biomax, and the quality and the resolution of the structures in different states and bound ligands enabled us to derive instrumental information to explain the mode of action of the enzymes.